Hello, welcome back to What is a Biology? I'm your host, Mr. Johnny Hopkins, and today we're going to be talking about something a little bit more interesting. It's called translation. This is not like going from Swedish to English. This is something a little bit more widespread and translated in every single human being in the world. It's going from mRNA to proteins. So this is the second part of the central dogma, if you remember that. So what does this is a thing called the ribosome. The ribosome, it's a tricky little thing. It's this fanciful RNA protein complex. So it's made up of two parts. So first of all, I want to say that this is the bacterial ribosome right here. The values of these S's and some other stuff are changed with the eukaryotes, but the ribosome of the bacteria are most studied because they're simpler, so we're going to focus on that one. So it has a 50S subunit and a 30S subunit. Now what the heck is this S? That S means Svedbergs. What's a Svedberg? It just means when you do some centrifugation, you spin something around really, really fast, and the rate of dissipation that it falls down to the bottom is Svedberg. So that's what this is, because they just separate it out using that. So that's how the terms work. So yeah, a large subunit and a small subunit. Actual nomenclature right there. So the small subunit is the 30S subunit as well. It's made up of the 16S rRNA. rRNA is ribosomal RNA. And 6 proteins. Large subunit is made up of 30 proteins and 33S. So coming together, 30 proteins plus 33S make 50S. But one thing to note is that addition of this, the 30S and the 50S, those guys combining together makes a 70S ribosome. So it's not always just a simple addition to this fanciful Svedberg unit. So here's the dealio. The good old dealio is that you have an E, a P, and an A site at the ribosome. 3 sites for tRNA to come in. Translational RNA. And so what it does is it grabs onto mRNA and it pulls it along, going from the 5' to the 3' direction, chugging along and making some protein. So it's got an error rate of about 1 in 1,000. So that means if you have 1,000 amino acid long peptide, then you have one of those residues being incorrect from the given mRNA sequence, and has an elongation rate of about 6 to 20 amino acids per second. So it's decently quick. So now, how does it make it? How does a ribosome be like this mRNA right here, this little fancy guy? What are you telling me? And it's got to translate what is in this mRNA sequence and make it into a protein, so it can have that protein go and do its job and make us think about applesauce all the time. So how it does this is something called a codon. There are codons, and these codons are what make up the mRNA. So a codon is just a 3 nucleotide sequence, and that's how it reads. So what this mRNA does is it just reads constantly from the mRNA, these triplets, and that's able to turn it into a new amino acid, which can translate it into a protein. So there are, so it's 3 nucleotides. That means we have 4 possible bases for mRNA, and that means raised to the 3rd power. So that means you have 64 possible codons. And there's only 20 possible amino acids, so you have a lot of redundancy, which help, but not too much redundancy. And we have one start codon that is always conserved, that is AUG, which encodes methionine. If you remember, it's a long, lanky boy with the sulfur in it. And then you have 3 stop codons, UAA, UGA, and UAG, and those guys stop the translation of the protein of interest. So, for example, you also have these codons of GCC, which goes to alanine, and CAC, which goes to histidine. Now, real quick before we get into the actual stuff, we need to also talk about tRNA. So we know rRNA catalyzes the creation, but tRNA, this is a fancy guy. So tRNA goes in and joins in one of these three spots and attaches its anticodon to the codon of the mRNA. So it's got this fancy secondary structure that you're going to see a lot and a lot and a lot. And particularly, it's important to note that it's got a 3' end right over here. That 3' end ends with the sequence CCA, cytosine, adenine. And with the OH, that's the acceptor stem. So that's where the amino acid gets added. So when you have an amino acid attached to this guy right here, then it becomes amino acyl tRNA AA. So where we just replace this AA with the amino acid 3 label thing. So alanine, ALA, tRNA, ALA. And so then it gets added there, and then it can go and join in and have some fun at the party. And 3 dimensions, one thing to note though is that this structure is not looking at you like, so it looks like this weird L. It's just kind of like, wow. You know, it's actually a lot like that, and you'll be surprised if you look it up on the interwebs. So, the mRNA makes a codon, the tRNA makes an anticodon, and how they sense this is by using the Watson-Crick base pair for the first and second position. And because there's a lot of redundancy, because we have 64 nucleotides, 64 possible codons, the third one is not as particular. And so that's where it comes in as a wobble pair. Wobble pair means that it's not as specific, but it's still a little bit more specific. And it helps reduce the redundancy and reduce the amount of tRNA needed to make these proteins in the cell. So one typical wobble pair is the GC wobble pair. So they have a G and a C kind of just move there's GU wobble pair. G and U kind of just move out and about and accommodate a non-Watson-Crick base pair. And then you have it, also adenine can be made into inosine, which is this guy shown here, which actually helps base pair with multiple different ones. So you have better cognate structures. One thing to note is that there's cognate versus non-cognate. A cognate structure means the anticodon and the codon actually Watson-Crick base pair and do a good job. But then non-cognate means that they don't agree, they don't bud. Near cognate means like one or two of them base pair, but the interaction is not very strong. And so the interactions of these Watson-Crick base pairs, this energy helps drive formation of the protein. So now, how does this happen? We can actually stop dealing with all this structural mumbo jumbo and get into the process. You got 3 steps: initiation, elongation, termination. So let's get started. Initiation is brought up by initiation factors. Initiation factors 1, 2, and 3. So as I said last time, factors are proteins that bind to nucleic acids. Okay. And one thing to note is that mRNA, we have a little bit extra usually at the end of the mRNA. And in bacteria, there's the Shine-Dalgarno sequence, named after some guys named Shine and Dalgarno. And so what happens is that actually IF3, initiation factor 3, will go and grab the 30S ribosome and bind to the Shine-Dalgarno sequence. And so then the ribosome will the 30S will kind of just go on along toward in the 3' direction, trying to find AUG, the start site. Then, once it finds that, then IF1 comes in and binds to the A site. And then we have F-methionine, tRNA F-methionine bind to the P site because that is the codon for the start. And then IF2 and GTP comes on in and joins. And then with some GTP hydrolysis, it pulls in the 50S, and you have the full 70S ribosome with the initiation factors dissociating with the proper initial tRNA at the right position. So essentially right here, you have the 30S joining with IF3 and IF1 and IF2 with the mRNA in there. And then we have the final structure with tRNA right where it's supposed to be. Now, we have the next part of the problem, elongation. So we got the first residue in there, we got the first amino acid. How do we get everything else in there? It's going to be a little difficult process, don't you think? And so here's how it goes. It's elongation and it's led by elongation factors. Who would have thought? You have, instead of being the typical 1 and 2, 3, like initiation factors, it's TU and G. And these guys are GTPases. So they take GTP and they use the energy of GTP to make sure things go right, make sure the guide elongates. So we have a tRNA in the P site, and then what happens is you have a potential tRNA with EFTU right standing by it with some GTP, and so what happens is it goes and it attaches to the mRNA, makes sure and tries to see, is this the proper mRNA codon? Are we in the right codon, anti-codon stuff? And so it kind of gets accommodated and it kind of gets joined in there in the A site with the current peptide in the P site, and then EFTU fits it in there, and it's pretty much in there. And then we have, and then when we have it dissociate, we can either have accommodation or rejection. So it gets rejected if it's not proper Watson grid cognate structure, and it gets rejected. So then it just kind of repeat the process over until the right tRNA comes and joins in and makes sure things are going right. And then when things do go right, you have accommodation, such that the tRNA comes in and it binds to the cognate structure, and they have an amino acid tRNA in the A site and a tRNA with the nascent peptide chain in the P site. Then EFG comes along, some GTP, hydrolyzes that GTP and moves the A site to the P site and the P site to the A site. And in the process, adds that amino acid from the A site tRNA to the joint chain. So then it keeps joining, ligating, getting longer and going through this tunnel. One thing to note about this little tunnel that in the ribosome is that it's quite small, and it doesn't allow for a lot of formation to happen inside the ribosome. So usually what happens is right once it emerges, then the protein starts to fold and fold and fold. And it's usually assumed that beta sheets cannot form inside there, but alpha helices, some can. It's still a major point of contention. So now we have this concept of ratcheting. This is not quite so intuitive. It's so the 30S ribosome, during this process, it'll rotate kind of like this, but it's not a lot with relation to the 50S. So rotate down a little bit. But this ratcheting does not actually mean that there's a single direction of motion. It doesn't mean that it's unable to go backwards, because there have been a lot of experiments that shows that you can get frame shifts and such with the ribosome, such that it actually can kind of go backwards. So this doesn't mean it's always forward precessive motion. It's just that it means that there's a precessive motion of the 50S and the 30S rotating, kind of like a ratchet, where you pull it up, down, and back up, down, back up. Now, one thing that is commonly studied as well is that you have these things called hybrid states. So during elongation, when you move from the P to the A site, from the P to the E site, and the A to the P site, is that you have hybrid structures such that during the process, the tRNA will occupy two different sites at once, such that when you go from the P and the A, they'll kind of join in with the P and the E and the P and A, and then with EFG, they'll go along. And one thing that I think I forgot to note is that this E site is the exit site. So once it gets there, the tRNA, it kind of just flops down and does whatever it wants, and goes back and gets some amino acids added to it by some random enzymes, and joins the process all over again by going to the A site if it is needed. Now, termination. We got to end this thing. So, this termination is caused by release factors, specifically release factors 1, 2, and 3. So, we have a structure like this as shown here. So we have this guy. And then we find a stop codon with this nascent peptide chain. So now this peptide is finished. It can go out into the world, stop being a wiggly boy, and start doing its job. And so it forms this complex structure as previously discussed. So ribosome finds the stop codon. So what happens is release factor 1 or 2, these guys are sequence dependent, will bind to the stop codon. And when you have the stop codon bound, or if 3 and GTP join in, and they attach to the A site, and then ribosomal release factor, RRF, comes in and joins the A site, while the protein escapes and then starts doing its job, goes to some chaperones, and does some really good stuff in the cell. And then we go along, EFG comes in and helps out and processes, and then the 70S is kind of destroyed now, with the 50S leaving. And then eventually the 30S and dissociates from the tRNA and the mRNA and becomes an empty 30S. And then the process can begin all over again with the exact same machinery. So that is, in a nutshell, the translation. So a few things to just remember that come in handy is that you have polysomes. Polysomes are just multiple ribosomes on the same mRNA. So you can just have multiple of these ribosomes just stacked on top of each other. So you have many, many different peptides being made at the same time. So you can make multiple proteins, multiple of the same protein from the same mRNA. So you can get a good immune response instantly within a few minutes because of the elongation period. Now, puromycin. Puromycin is an antibiotic that is extremely used in things like ribosomes. And this antibiotic, it results in premature chain termination by mimicking adenine 76. So right here, this A is adenine 76. And it looks a lot like this, and it's got this, it's got something that looks like an amino acid at the end. And so what happens is it joins in at the A site, and it can't really do anything. And it just kind of, the ribosome just kind of is like, I don't know what to do, I'm just going to attach this and can't go on any further. So it escapes and the cell dies. So that's one type of antibiotic. Not all antibiotics work like this. It's just one specific type, puromycin. So, we're gonna go just do a real quick summary. Real quick! So, translation, it's done by the ribosome, which has E, P, and A sites, with tRNA going from goes from the A to the P to the E site and exits by matching with the codons in the mRNA, because the tRNA has the codons. The ribosome goes from the 5' to the 3' direction of the mRNA. tRNA has that little fancy structure. The codon-anticodon pairs are done by Watson-Crick base pairs, with the wobble pair in the third position, which can be done by a GU mismatch or an A or adenine to inosine. Initiation is done by some initiation factors that essentially grab the ribosome at the Shindo-Garno sequence, and they grab the 30S, put it on there, and grab the 50S and make sure everything starts right good with the methionine at the right position at the P site. And then we have elongation, where EF2 comes and adds on a cognate tRNA, and then EFG take it and move it down one, with some hybrid states forming in the middle. And then termination, release factors release the final protein that is supposed to emerge, and we have proper function. and then eventually the ribosome dissociates so it can all begin again. And that is everything. So that is everything by translation in a nutshell. Thank you for watching.